First of all, well, my name is Antonio Lamarca, and I'm one of the two co-organizers of this meeting. I'm professor of obstetrics and gynecology at the University of Modena, this city. In reality, we are here in Maranello, which is a very famous city because of Ferrari car, of course. And we are very close to Modena. And I'm also chairing the OBGYN department at our university hospital. So again, thank you for being here. And a special thanks to the faculty, which is made of worldwide recognized speakers. I'm pretty sure we are going to have today a very interesting and useful meeting, which is focused on ovarian physiology, ovarian folliculogenesis, with a look at the clinical application of this knowledge. So again, thank you for being here, and I should thank Medea, Chloe, Martapani for the wonderful organization. Let me introduce what Medea is. Okay, so Medea is a global medical education provider. It is dedicated to developing, implementing, and evaluating medical education programs worldwide. We have here the therapeutic areas covered by the Medea, female and male infertility, endocrinology, cardiology, rare diseases, and neurology. And you have here the panel of experts dedicated to female infertility. Robert Fisher is our director, and then you can recognize many colleagues and experts. Some of them will be here today with us. Then we have male infertility with Sandro Estives, artificial intelligence in reproductive medicine and biology, again coordinated by Sandro, reproductive biology, and fertility nursing and midwifery. Now it's a pleasure for me to introduce my very good friend Livio. Livio, first of all, Professor Livio Casarini, first of all, is a fantastic person. And after that, he's a superb researcher and professor. Well, he's recognized because of the long list of publications on ovarian physiology, granulosa cells physiology. And it's a pleasure for me to collaborate strictly with him and his team. We are learning so much each other, I suppose. And so please, Livio, the floor is yours.
Thank you very much, Antonio, for this very kind introduction. You know that your nice words are reciprocated. Well, first of all, let me thank my esteemed colleagues, my co-organizer, Antonio Lamarca. He doesn't need enough introduction, but he will be, of course, introduced later when he will have his talk. But also let me thank our faculties, Professors Carlo Alvigi, Baris Atta, Francesca Klinger, Nathalie Di Clemente, Renato Fancin, and Stine Christensen. Well, I would like to remind you that you may listen this conference using the podcast. So please connect to the Medea website to check the podcast. And also we are on live session now. So we have guests that will be introduced later for this. And also we can try the free live streaming always online. So this is the faculty disclosure slide. In our agenda, I think that we should be proud to have a lot of esteemed and world international experts in the field that will compose our fourth session of this meeting. In the session one, We will talk about new insights from basic science. I will not go into each session because the chairs will do that. In the session 2 this morning, we will talk about what clinical evidence is telling us. While in the afternoon, the first session is new insights on folliculogenesis and ovarian function, and the last one, future perspective for ovarian treatments. So I hope you will enjoy the meeting. Let me thank again Medea and our partners that are several scientific societies. I cannot mention all because the meeting should go on, but this is a thank you for you, as well as for other institutions, especially the University of Modena and Reggio Emilia and Merck for the support. This is a reminder for you. Please fill the press survey. You should have received a link, or anyway in the chat there is a link that you could use to reach the press survey. And this is a conference where you can obtain the accreditation, so fill out the post-event survey because you can receive by email your accreditation, and all the explanations are online. Let me say that in this conference we have a lot of people in this here attending in this room attending the conference, but also we have a lot of participants that are around 350. They are online, so welcome also to you. Among those, we have the 56% that have more than 10 years of clinical experience in the field, but also the 44% have up to 10 years of experience. So we are really proud and hope that this conference will provide helpful information for you. One of the last advice for you is please use the Q&A section in the online panel to put your question, and the chairs will select those questions to be delivered to speakers. Of course, they have to select them because sometimes they are a lot, but they will do the best to cover a wide field. So I think it's all. The meeting could start. So I'm pleased to introduce the next chairs, Professor Steen Christensen and Professor Antonio Lamarca.
So introduce Francesca. Yes, please. Okay, with great pleasure, I give the word to Francesca Klinger. Francesca is professor in histology at San Camillus International University of Health Science in Rome. Francesca has a broad background in reproductive and developmental biology, in histology and stem cell biology, with a primary focus on investigating how chemotherapy treatment can impact on subsequent female fertility and ovarian function. So please, Francesca, the floor is yours.
Okay. Thank you very much. Thank you, Antonio. And thank you, Livio, for this very kind invitation here today. Good morning to everyone, first of all. And thanks also to Medea for this wonderful workshop that we have today. First of all, so I'm here and I will, as Livio said, take you in basic science. I apologize straight away if I go too much in details, but I try not to go too in depth in all the pathways so that you can easily follow me. And what I would like to introduce this morning is how we can modulate the ovarian reserve, how we can promote, as is written in the title, quiescence in some way or waking up these follicles. So to do this, I will go back and I will start my presentation with something that for sure you all are very well aware, that is how the follicles start from a primordial follicle pool that we have in our ovary, and then they can be either, as you can see, recruited, they can be maintained inside these pool as primordial follicle, or sometimes they also go through atresia. All these processes are maintain in a sort of balance, so that we have always, until menopause at least, some primordial follicles that will be maintained as primordial follicles, while all the others will start the recruitment and the atresia. What do we know about this dynamic? This dynamic, we know now that is activated in some way, so that we have pathways that will promote the recruitment, such as the PI3 kinase pathway. Then we also have the Hippo pathway that will promote the activation, and on the other side we have factors such as the AMH that maintains the pool as primordial follicles. There's another group, let's say, of studies that show us instead how these follicles can be brought, let's say, to atresia or to apoptosis. And in these years, we have focused our attention, for example, on the apoptotic pathway, elucidating the role of TAP63 alpha, that is a protein of the p53 family, that controls the quality of the DNA of these oocytes. As soon as the quality is not the best, the apoptotic pathway is activated by this protein. So this is something that goes normally in a physiological way. We have some so that will be maintained in the reserve, some that will start the recruitment and then the maturation, and others that goes in atresia. Of course, we can have unbalance of this situation. And a typical imbalance is when we do have an extreme loss of these primordial follicles that leads to what we all know as being premature ovarian insufficiency. Several are the causes. I will not go through all of them, just to point out instead the importance of, for example, the chemotherapy. Chemotherapeutic treatments will decrease very rapidly, sorry, this is the curve, of the amount of these primordial follicle, inducing what we know apoptosis. So this is where we started to work on, based on these information, and in search of what we now call a fertoprotective adjuvant, something that was promoting the survival of the primordial follicle. We tested the possibility of LH to be one of these fertoprotective adjuvants. We started with experiment in a mice model. So we recapitulate in the mice model the premature ovarian insufficiency with two chemotherapeutic drugs, cisplatin, and in this part of the results, cyclophosphamide, that are two very common chemotherapeutic drugs used. So what we have done was initially an in vitro analysis of our follicles. What you see here in green are the oocytes that we are able to follow in time, and if you check the small ones, when they are treated with cisplatin, they disappear after a certain time. So instead, they do not disappear, let's say they do not go in apoptosis when we give LH. These results were also recapitulated in vivo, so giving exactly the LH to these animals that had a reduced fertility due to the chemotherapy. And the same results were also obtained, so with cisplatin and cyclophosphamide. What is then the mechanism? How does it actually work? So, and especially, are these oocytes of a good quality, those that we protect? The LH is able to counteract the effect on the DNA damage in red, and so very few oocytes are left with cisplatin, and they are all damaged. The red spots indicate the damage of these oocytes. With LH instead, we rescue these oocytes and they're not damaged. Similar, let's say, effect on the DNA is also when we observe the alignment of the chromosome that is they're well maintained in the presence of LH. What do we know also is that the LH receptor is not expressed by the oocyte itself. So how is this LH acting on these primordial follicles? We did analyze the effect also of the somatic environment, so what was present, and in particular, I want to point out that all the experiment, it's something that probably I skipped a little bit, is that we are working mainly on pups that express, that have in the ovary only primordial follicles, very few primary and almost no secondary follicles, okay? So the receptor for LH is present in these ovaries and modulates as well its action on the ovarian somatic cells that are present. I'm not going through all the information. Just for the sake of time. So and also modulates the environment. We have analyzed how these the proteins expressed in the environment of these ovaries are modulated. After LH, we do the treatment with LH, we see an increase of anti-apoptotic proteins, an increase in cell proliferation markers, and of course a low reduction instead of apoptotic proteins. So this is the results from which we started on mice, but of course then the following question would be, okay, is all this can be recapitulated in humans? So the experiments that we have performed was on the human ovarian cortex that was fragmented and again, as you can see, treated here in culture with cyclophosphamide and LH. After the culture, we have analyzed these cortex. We checked the follicles, and what we have seen is that again, our attention, our focus are always primordial and maximum primary follicles, also because in the cortex, that's the population that we see, okay? So cyclophosphamide is inducing a reduction of these follicles, while with LH, we do see a rescue, again, of primordial and primary follicle. Chemotherapeutic drugs is inducing DNA damage, and LH is reducing this damage. What we have found is that it does not prevent the damage, okay? LH is able to, it sort of gives times to these follicles, to the oocytes, to repair the DNA. It does not prevent completely the damage. And again, even in these cortex, we analyze the expression of the receptor, either by Western blot or by immunohistochemistry. And again, we did find that the receptor is actually expressed even in these cortex where there are no theca cells, or at least we did not find the theca cells, or any antral follicles. The LH finally promotes also the proliferation of the somatic cells in these cortex. So what we can conclude for this, let's say, first part of our results is that even in human cortex, LH is able to reduce the DNA damage, to reduce apoptosis of the oocytes, to promote DNA repair, and again, I have not shown you these results, but counteracts the pro-inflammatory microenvironment, and these inflammatory reactions are caused by the cyclophosphamide. But while we were doing all these experiments, Antonio has published this wonderful paper that prompted us to investigate now a little bit more other effects on LH. He did find that an extended LH administration could be a strategy to increase the pool of recruitable antral follicles. And also we did know from Livio's experiments that LH activates those pathways that are known to promote the activation of the follicles. So from going on a, let's say, the POI model, that is when we induce a damage, so we are inducing a depletion as a pathological condition. Our question was, sorry, anyway, I go to the question and then I go back. Our question was, can LA play a physiological role in the activation? Is the receptor is already there? Could this receptor be functional and actually work in some way? So at this point, we need to go back a little bit and see which are the pathways and how the relation between the oocyte and the follicular cells can be modulated in terms of the activation. So we have worked, let's say, on the maintenance since now and against the atresia. Now we want to see this line here, the recruitment induced by LH. What is known is that the oocyte itself can activate these pathways for when growth factors such as, example, KIT ligand binds to its receptor and so promotes the activation, but also signals from the follicular cells can induce the activation of the follicles. So when we are now back on our mice model, is the easiest way for us to work and then to pass to human samples. Again, our model of pups, where we do have only mainly primordial follicles, and even on the somatic cells. What is the effect that we have seen in vitro? Just focus on this image, graph here, where the pink line shows us an increase in the size of the ovary, of the oocyte in the presence of LH. So now we are just giving LH, no chemotherapy, and just giving LH, and we do see that these oocytes become bigger. A good thing of our culture system is that we can follow every single oocyte. The problem is that it's a 2D culture and it has some limits. At the moment, the best timing is up to 120 days, so around 4 or 5 days. Afterwards, the culture is not anymore brilliant, so we have to move to something different. On the somatic cells, we have seen that giving LH promotes the itself the expression, so there is an increase of the expression of the receptor and an increase in the expression of the KIT ligand. So we are actually stimulating the production of KIT ligand and the proliferation of the somatic cells. We moved to the in vivo studies at this point, injecting directly LH on two models. So this time we went for the pups, as I was saying, so promoting just the primordial follicles or acting on them. And on the other side, what are young adult, that means just after puberty, so once they still have primordial follicles, of course, but to see what would be the effect later on. After what are considered like two estrous cycles of mice, we collected the ovaries and we checked several things. One is the volume of these ovaries. The first thing, if there is a growth of the follicle, even the ovaries should grow. These are the experiment on pups. These are images in which we can see the whole ovary, and we do clearly see an increase in the size of these cells. So then we went back to a classical histology instead to check the follicles. And in this case, this is the whole count, but for simplicity, the two main follicles that I want to point out are the primordial, that you see with LH are even a little bit higher than the control. And on the other side, the atretic follicle that we start seeing are decreased in the presence of LH. Sorry, because it's a little bit different, so if I see here. Then on same type of experiments on the young adult, again, increase in the volume of the ovary. We are now checking with this technique also the count of the follicle. It's only that the procedure is a little bit more complicated than the normal histology. And so for the normal histology, went directly, we have the whole follicle count, where we now have their young adult and fertile female. So we do see all the stages of the follicles, secondary, antral, and even the corpus luteum at this stage, with the atretic follicles also here. And in this case, we see again, you see an increase, or anyway, a survival of the primordial follicle higher than in the control, a decreased amount of atretic follicles, but the new thing that we see, even the percentages are very low, but the mice are still young, the presence of corpus luteum when we treat with LH, something that instead in the control is not present. These results are confirmed even by the assessment of an estrous cycle in the pups, and the let's say LH mice, as we call them, are all in estrus, indicating, so in this case, that anyway we are sort of accelerating the something that anyway happens physiologically. So in this part, we instead can conclude that there is a promotion of the activation of the follicles, with the probably again an increase in the expression of the receptor and KIT ligand. It limits atresia, both in pups and young adults. And what we think, since we have seen folliculogenesis stimulated more in the young adult than in the pups. The LH, yes, is stimulating, it's accelerating something that happens physiologically, while in pups, what it's doing is promoting a better environment for the primordial follicles. So when we give the pups that have only primordial follicle, these follicles are healthier, let's say, and so numbers are even higher than a normal control. When we give them to young young mice, instead we are accelerating folliculogenesis. And so with this, I would like to thank you for your attention and thank the group. We have the students that have worked on this project, Valerio Rossi, Serena Marcozzi, Harpreet Lamsira, and now Nicole Bertani. So it's a long story through PhD students. Thank you very much.